Hey guys, it's Mr. Wolf here, and I'm going to be going over meiosis again. This time we're going to have 3 sets of chromosomes, or 6 chromosomes in my cell. So let's talk about the purpose of meiosis again. Remember, it's to make our haploid gametes, our sex cells, whether they be sperm cells or egg cells, and we want them to have genetic variation. We want them to be different from one another. So this is our goal of our resulting cell. So how are we going to accomplish that? Well, we start off with these homologous chromosomes in our cell. All right, one from mom, one from dad, and they're going to have the same genes. However, like they might have different versions of the gene. While this one might have these might both be the gene for hair color. This one might be brown. This one might be blonde. Okay, and this cell is going to be a diploid cell, because it has 2 sets of chromosomes, right? I have 2 of the long chromosome, 2 of the medium, 2 of the small. All right, so how this process is going to go down is we're going to first do DNA replication. So let's get the diploid out of here, and all my chromosomes are going to make copies of one another. Okay. And at this point, technically, my cell would be a 4N cell, which all it means is that it just has 4 sets of each chromosome. 1, 2, 3, 4, the long ones, and so on and so forth. Okay. Now, the cell's going to split, but before it does that, we need to do 2 things that are really going to help in creating this genetic variation in the long run. And the first one is going to be called independent assortment. And all that means is just how these chromosomes decide to line up. Let's say they decide to line up like this. It's completely random. So in this case, maybe we have just by random chance, we have all the yellow on one side, all the red on the other side. But it could have just as easily happened where maybe the red right here lined up on one side, and the red lined up here on one side, and then the yellow on the other, like this. Right? And so when this happens, you can imagine when the cell splits, you could get different combinations each time depending on where your chromosomes are at. All right, so that's our independent assortment. It's going to help with genetic variation. The next thing that's going to happen is going to be our synapsis, or crossing over. So once the cells do line up, they're going to swap parts. So here I'm going to have the top of my chromosome here, swap parts with the other half. Same thing right here. And the same thing right here. Okay. So if I just look at my chromosomes right now, not one of them is identical to the other at this point. Okay, but I still have a cell that has 4 sets of chromosomes in here. So that's twice as much as a normal one. So what needs to happen is this cell needs to divide. So let's kind of get rid of the cell membrane right here, and these chromosomes are going to move over. These chromosomes are going to move over like this. And as a result, we're going to get 2 new cells. Right? And at this point, we're back to our diploid cells. Right? However, we have accomplished a lot of genetic variation at this point, because my cells are not identical. They look different. Okay, the next thing that happens, since our goal is to make these haploid cells, is we're going to split one more time. So let's get rid of these cell membranes one more time. Now keep in mind the cell membranes actually stay intact. They just kind of move apart from one another. But I'm going to move the chromosomes here, here, and here, and then these ones are also going to split. And I'm going to have kind of these weird looking cells just because I don't have a whole lot of room on the screen. And so what I'm left with are these 4 cells. And these cells are haploid because each of them has just 1 set of each chromosome, right? Yeah, there's 3 chromosomes in here, but there's only 1 small one, 1 medium, 1 large one. Right? If I look at all these cells, I've created genetic variation. None of these look identical to one another. All right? And that was due to my crossing over and my independent assortment.